Today we want to talk about penicillin allergy, a somewhat advanced topic in immunology. And you see the penicillin up here. You see that is an antibiotic of this class of beta-lactam antibiotics. So you can see this beta-lactam ring here. And we want to talk about the allergy to this penicillin. And often this includes additional penicillin-like antibiotics, penicillin derivatives. However, generally not all of the beta-lactam antibiotics, but it can in some cases also extend to other beta-lactam antibiotics of other classes. It's relatively frequent, especially it's reported as relatively frequent, even if in practice maybe it's not as frequent as it may seem. We have seen a table similar to this in microbiology already. This is kind of only an excerpt of a bigger table, but what you see here is different antibiotics. And then up here you see different bacteria, and both could be extended further. So we could still look at more antibiotics and more bacteria. And then the general idea of this table is a bacterium susceptible, or is a antibiotic effective for a given bacterium, in which case this field would be indicated in green. And you can see that for most bacterial species you have more than one antibiotic that you can use. And this is important for several reasons. So, for example, over here you see the route of elimination of this antibiotic. And for people where this organ is damaged, you may not want to use this antibiotic. Or in some cases, the antibiotic is damaging for certain systems or for certain parts of the body. So, for example, we have ototoxicity for some of them, or some antibiotics are harmful for pregnant women. So you have indications where you just can't give this antibiotic. You have the case that certain antibiotics work well for infections in certain organs, but not in others. That also has to do with mode of application, for example. So many reasons why you want to have more choices than just one generally. And then one additional big reason is these allergies, as for example the penicillin allergy, which, as I said, is relatively frequent. Okay, we want to start by just revisiting what we have learned before. So just think about what are the 4 types of hypersensitivity reactions. So just take 2 minutes or so to go through those. Okay, then we will continue with the next question. And the next question is, what is an allergy? And I'm not just looking for the definition here, but also describe what happens at the cellular level. Okay, good. So to go for something like a definition for the second question. So an allergy is a strong reaction of the immune system to a harmless foreign compound. This is to be set apart from autoimmune reactions. There you would have a reaction of the body to a self-compound. But allergy, it is to a foreign compound, which is what the body should react to. But in an allergy, this reaction is stronger than it should be, or strong in a case where there should be no reaction at all, because this compound is not a pathogen or anything. It's not something that's going to harm us. With regard to the 4 types of hypersensitivity reactions, I don't want to go through all of them in detail now, but they're shown here, so you can revisit this slide later on. What I want to look at now is this type 1, so the immediate type reaction, which is the one that's the most relevant for allergies. And just to recap, what's shown here is a mast cell. And this mast cell has these IgE antibodies bound to its surface receptors. These IgE antibodies are built upon the first contact with the antigen, which in this case is the allergen. And then upon the second contact, once these IgEs are attached to this mast cell, this allergen can bind here, and this triggers the degranulation. So these substances are released from the mast cell, and the most well-known of these substances is histamine. And this has these consequences, which we then know as the allergic symptoms. So these are compounds that lead to the widening of blood vessels and also increased permeability of blood vessels. And consequence of this is, for example, redness and swelling and pain. Okay, next question for you to think about. So we have seen these antibodies before, and now the question is just how do B cells get activated? Again, take 5 minutes to think about this question. Okay, how do B cells get activated? We can do this review together in more detail here. So let's start with a antigen, which is detected by a B cell, so by this B cell receptor, which is essentially a surface-coupled antibody of the IgM or the IgD class. And this B cell will now take up this antigen and will digest it into peptides and present those peptides via MHC2 on its surface. What do we further need? So this is, by the way, a process that typically occurs in secondary lymphoid organs, so in the follicles of the lymph nodes, for example. What else do we need for full B cell activation? We also need this antigen to be recognized by a professional phagocytic cells, so dendritic cells. Typically this happens or can happen in the periphery of the body, and also this dendritic cell is now going to take up this antigen and present a fragment on its surface again via MHC2. The dendritic cell will now go also to a secondary lymphoid organ like the lymph nodes, and here if it's a if it's a foreign peptide, this may be recognized by T helper cells via their T cell receptor. There will be additional signals here, so additional membrane-bound molecules that will interact, and also cytokine signals, and overall this will lead to the activation of this so far naive T helper cell. And now this activated T helper cell can bind the same peptide, which is presented via MHC2 by the B cell, also a reaction that happens in the lymph node. And this contact, again with additional contacts and with cytokines, is the one that leads to the activation of this B cell. Also further proliferation of this T helper cell, so activation and proliferation of the B cell, and among others, maturation into a plasma cell. Plasma cells are often shown with this very predominant Golgi apparatus because they produce a lot of proteins, and the proteins that they produce, of course, are antibodies. And now, as we have seen before, these antibodies can bind to the surface receptor of these mast cells. This is if those antibodies are of the IgE class. So during this interaction of T helper cell and B cell, we also have a reaction known as class switching, so different forms or different classes of antibodies can be formed now. And for allergens, often we have this switching towards this IgE class, which is the one that can bind on the surface of the mast cells. And now, if there's again a second contact with the allergen, then as we've seen before, we have this degranulation of the mast cells and the release of histamine, for example. Okay, something important that I want to draw your attention to, we had this before already. What is recognized here by the B cell can be one epitope, so which is shown in red, and typically this is a structured epitope, so these antibodies recognize structure. But what is then presented via the MHC2 can be, or often is, a different epitope, so which is the one that's shown here in green. And then up here at the dendritic cell, we would have the same epitope, so that we then can have this T helper cell activating this B cell, which also presents this green peptide. And then again, but this is going to lead to the stimulation of this B cell and the formation of antibodies, which again recognize this red epitope here. So the one that's presented by the B cell and leads to stimulation can be and often is different than the one that's actually recognized. So now thinking about this B cell activation and this maturation of B cells to plasma cells in the context of penicillin allergy, we have a problem though, because penicillin, as we saw on the introductory slide, is a small molecule. It's not a protein. So the binding of this small molecule to the B cell receptor, that's not the problem. The binding of a small molecule to antibodies, this is not the problem. So also if we develop antibodies for use in the lab, we can develop them against small molecules as well, not just against proteins. The problems come in here at this presentation via MHC2, so both on the dendritic cell and by the B cell, because what's presented here is peptides. So this is a protein that's digested via the proteasome into peptides, and then these peptides being presented. And obviously this can't happen if this one is not a protein, if this one is a small molecule. What's the solution? How can penicillin allergy still happen? How can we still have this maturation of B cells to plasma cells against penicillin? Well, this is the case if penicillin gets bound, and usually covalently bound, to some protein, and then what the B cell recognizes here and what the antibody eventually recognizes is this complex between penicillin or a penicillin derivative, like something that's partially metabolized, in complex with this protein. And then, in theory, the process could happen here that, this part where the penicillin is bound to the protein is recognized, but then what the MHC molecules need to present is only another peptide, as we've just shown before, another peptide of this protein, and then we can still have antibodies that are directed against the penicillin, which is linked to these proteins. I said in theory, because next I want you to think about this question, or about think back to your previous knowledge, what you learned before. The question is, what is central tolerance? And again, take a couple of minutes and think about this question. So, central tolerance is the mechanism by which our immune system avoids recognizing our own proteins as foreign. And we're not going to go into detail of the mechanism now, but this has to do with this so-called school of the thymus. So in the thymus we have these T cells which are eliminated if they recognize self-proteins via this process of negative selection that we talked about before. So how does this fit in what we're talking about here? Well, we said the penicillin is bound to a self-protein, so typically this is albumin or some other abundant protein in our body, but it's bound to one of our proteins. And then we said, okay, the mechanism how this B cell activation could still happen is if what we have then presented here is a peptide of this protein, but again, this is one of our own proteins, so this should not happen if this mechanism of self-tolerance indeed exists, which should exist in a healthy immune system. So the question again becomes, how can there still be penicillin allergy if this shouldn't occur? And the answer again is relatively simple. And the answer is, well, what is actually presented here is not any peptide, but is actually the peptide that is linked to this penicillin, covalently linked to this penicillin at those different points here as well. And now it's this penicillin peptide complex that's presented here. And this is something that doesn't occur naturally in our body, which is not one of our proteins that the T helper cells would learn in the thymus that they are on our own, because it's now this modified peptide, this penicillin modified peptide. So again, the B cell recognizes this protein peptide complex. It's this protein penicillin complex. It's this peptide linked to the penicillin, which is presented here. Same here. So in this case, the dendritic cell also needs to take up not just the endogenous protein, but the penicillin protein complex. Present also this peptide that is covalently modified with penicillin, and then this T helper cell can recognize this as something that's non-self, so as a foreign substance, and can therefore activate this B cell, and the rest of the process is the same. So we have maturation to plasma cell, production of these antibodies, and then degranulation of the mast cell upon a further encounter with this penicillin protein complex. So this now is the mechanism by which we can have penicillin allergy. Important term to know here is the term of hapten. So hapten is a substance that can be immunogenic, but only if it's bound to a protein. So it only becomes immunogenic if it's bound to another protein. And this is a mechanism that we often use for vaccinations, for example. So if we want to make a vaccine against something that's not a protein, so let's say a bacterial capsule, then we would already in the vaccine covalently link this substance to a protein so that it can be immunogenic in a way that antibodies are formed. In this case, of course, hopefully not IgE, but other antibody classes. Okay, just a couple more things to go more into depth in this topic of penicillin allergy, even so the fundamentals we have understood now. What I showed you here was a type 1, an immediate hypersensitivity reaction. The type 4 reaction, the delayed type reaction, might also play a role here. So in this case, this part is not relevant, but it would be relevant that we have these activated T helper cells against this penicillin-bound peptide or penicillin-bound protein. And then it would be the other actions of these T helper cells other than B cell activation, which are relevant. So, this other cytokine mediated functions and so on, or these may not necessarily only be T helper cells which are stimulated, but also cytotoxic T cells which then exert their functions. And then, of course, an interesting question is, so if now this mechanism of penicillin allergy exists, or potentially also other small molecules, other drugs that can covalently bind to proteins, then why doesn't everybody who ever was treated with penicillin develop this allergy? So there are certain factors that influence this, and one factor is genetics, because these HLA are not the same between all of us. So there are groups of HLAs, but certain people have HLA from one group, other people from the other group, and depending on which HLA you have, you may be more or less likely that this happens. Also, genetics in terms of how drugs are metabolized in your body, because we said sometimes it's not the penicillin itself, but a metabolized derivative, which is the one that's then covalently linked to these proteins. Very important here, of course, as well is exposure history. So we saw both the macrophage and this B cell would need to encounter this modified penicillin. So and then you need not just one exposure, but then also the second exposure, so that the antibodies that are now linked to the mast cells again encounter the same protein. So you need a fairly high level of exposure and a fairly high frequency of exposure. So this is important, both the frequency but also the mode of application. So penicillin exists in a form that's applied orally, but also in a form that's applied via IV. or intravenously. And so the second one, of course, makes this more likely. Also, age seems to play a role when you're exposed to this penicillin. And then also, once you have a penicillin allergy, this can also fade again as you get older. As so often in allergies, the microbiome seems to play a role in a way that's not well understood. And of course, also just chance. So by chance, some people might develop this allergy, and some people with similar genetics and similar exposure history might not. One important thing that I tried to allude to already in the when we were at the introduction, the introductory slide, is sometimes people might believe they have penicillin allergy, but they actually don't. So the numbers are something like that people, especially if it's the patient themselves reporting if they have penicillin allergy, if they're just asked, do you have penicillin allergy, then about 10% of people might say, yes, I do have this allergy. But the truth is that's about an overestimation by tenfold. At least that's kind of the numbers you find in the literature. Often this is a misdiagnosis. Often it's side effects of the penicillin, like you get a rash or something like that, which people then might think, okay, this is an allergy, but it's actually a side effect. And it's a big problem because if your doctor assumes you have a penicillin allergy, then it limits his treatment options. And then he might resort to antibiotics, that are just have more side effects or have other problems, are more broadband, so it might kill parts of your microbiome that you wouldn't want to kill in addition. Or simply things that, like, you don't go to the first line, but you go to the second line antibiotic, which is not good in context of resistance development and antimicrobial resistances accumulating. Okay, let's recap from what we talked about today. So this is first for you to think about it yourself again, if you can remember, if you understood those things. So first question is, how can penicillin cause a B-cell response, even though it is a small molecule, i.e. it's not a protein? So pause for a little and think about this question. The second question is, how can a self-protein cause an immune response in the context of penicillin allergy? Okay, again, pause for a bit, and I'm sure you can come up with this answer. Okay, and to summarize the most important points, in penicillin allergy, penicillin acts as a hapten, and here is again what hapten is. So it's a small molecule that causes an immune response only when it's attached to a larger carrier molecule, i.e. typically a protein. And the penicillin-bound peptides are recognized as foreign, even though they are on a self-protein, but because it's what is recognized is this complex between the protein and the penicillin, or the peptide and the penicillin, this is something foreign that wouldn't be presented in the thymus when those T cells are selected. I should say this is importantly one mechanism in which penicillin allergy can develop. There are most likely other mechanisms as well. So for example, mechanism where the penicillin doesn't need to attach to one of these soluble proteins, but where the penicillin actually would attach to an HLA, to an MHC2. And then it would be this which would be recognized by the T cell receptor, and penicillin here may even work as something like a super antigen, so it cannot be recognized only by one subset of T cells with a certain receptor, but potentially even by many of them, so that you get a very wide response, so that you kind of indiscriminately activate T helper cells. And additional mechanisms for penicillin allergy have been proposed and described as well. So I've put one reference that gives a bit of an overview here.